Hi, so today, next 5 minutes, I will be taking you through why the XPS and NEXAFS spectroscopy is a powerful tool for in situ and operando mechanism study. So this is the outline of today's talk. So so this is the actual setup of the any XPS or X-ray technique. So the difference from the lab mm, to synchrotron radiation is the source. The source does changes many things. The signal contribution is given by this equation. The sample, mainly sample, and then the source. The detectors are same in both at synchrotron and then the lab source XPS. But there's this the source is different, so that's basically you have a bright light source here, so that changes many things. Allow you to high flux, allow you to do the ambient pressure studies, and also you can able to trace the small amount of element present in your catalyst or in your liquid. So the key advantage of synchrotron radiations, so you have a tunable photon energy that allow you to avoid the Auger electron. And sometimes the Auger electron give you a very useful information, local structure of your catalyst or catalyst electrolyte interface, and but changing kinetic energy, so you can make it more bulk to surface sensitive measurements. And so you have a high flux density, very focused and bright beam and it's wearable. It allows you to do the geometry studies, and it's also allow you to do the time result studies. So an X-ray absorption is a complementary to XPS synchrotron radiation. So XPS give you the field orbital information, XAS give you the empty orbital information about the conduction band. So including the Auger electron spectroscopy as a whole, you can understand. So you can understand full structure of the catalyst or local structure of the electrolyte and catalyst interface. So that's the major advantage. There are drawbacks like access limit and sometimes high flux potential potentially causes the beam damage. Yeah, so we have, this is our schematic of our insertion, our beam line at synchrotron radiation Diamond Light Source. So we have we use basically penny magnet and two different bays placed into two lines, and they work as two different beam lines and a few insertions. Particularly this insertion is an XPS insertion. Another one is the NEXAFS insertion. So these two beam lines can accommodate the high pressure cells and also electrochemical cell. So we are more interested in situ and operando and XPS and NEXAFS such as surfaces and electrochemical system, particularly electrolyte catalyst interfaces. So because these signals, the studies, this kind of studies will provide you key information on active sites and the reaction intermediates, and also the kinetic of your reactions, so that allow you to rationally design an energy efficient catalyst. That's for that reason we develop an electrochemical cell. There's a 3 electrochemical cell, so we can do operando NAP XPS and both and NEXAFS and XPS basically. And so, for example, one of the example we can study the hydration structure of your electrolytes in the system, any electrochemical system. And second example is you can You can study the redox chemistry. For example, here is a copper redox using the X-ray absorption spectroscopy, how copper, species changes on the surface as a function of applied potential. Yeah, so you can also, we have a light source on the end session. You can also do the photoelectrochemistry. So while probing in situ while doing the photochemical reaction. So that's one of the application. And as I said, we can also do the XPS. So similarly, so we look at directly at the surface during operando measurement and measure the as expressed surface sensitive. We measure the surface species in real time. So one of them is here, for example, the industrial catalyst iridium oxide, where we looked at different oxygen species and understand the reaction intermediates. Yeah, that's the brief, brief introduction about the XPS and NEXAFS synchrotron radiation. So these are powerful techniques in studying in real time of any electrochemical system. Thank you so much.